The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, best digital health, best incubator accelerator, best startup.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Well, welcome all to the Galien Foundation Week of Innovation webinars here in New York and all around the world. I'm Michael Ringel, Managing Director and Senior Partner at BCG, and I'm delighted to be your moderator for today. The objectives of this week is to provide you with background for our largest annual event, the Prix Galien USA Awards, held this Thursday here in New York, in conjunction with our annual Galien Forum of speakers representing top scientific experts and leaders in global health. So Galien has developed this new webinar series to give our nominees in 6 categories: best pharmaceutical, best biotech, best medtech, best digital solution, best startup, and best accelerator or incubator, greater visibility in describing their journey of medical discovery. This year's Galien USA Awards has a record 147 nominees from 126 companies, covering 15 therapeutic areas ranging from cancer, which we'll talk about today, to cardiology, to neurosciences, to infectious diseases and rare genetic diseases and vaccines. So we'll be looking closely at all the products in these critical areas throughout the week. This webinar, as I think everyone knows, is focused on best biotechnology. And because there are 21 products in total in best biotechnology, we've cut it into 3 sessions. This session is covering cancer. We have 4 nominees, which we're delighted to have here today. The central question we'll talk about is, can the foundational biological understandings that are now being brought to bear in cancer care be leveraged to secure even more targeted, even more personalized approach to other rare conditions, particularly blood-based hematological disorders, as well as existing cancers to which it's been applied so far. So in the hour we have allotted, we'll have some time for prompted questions, which I'll moderate, but we're also very conscious we want to leave time for open questions from the audience, which is always the most fun part. So please, if you have questions, forward them to us in the link that's provided on your screen. Okay, so let's get into our discussion without further ado. I'll start by introducing the 4 nominees, representatives who are joining us for this webcast. So first, delighted to welcome Gerald Meinhardt, Vice President of Asset and Portfolio Management at Daiichi Sankyo, nominated for Enhertu. Second, we have Lusang Liu, Senior Vice President of Innovation at BeiGene, nominated for Brukinsa. Third, we have Kristen Heggie, Senior Vice President, Early Clinical Development, Oncology, Hematology, and Cell Therapy at Bristol Myers Squibb, nominated for Abecma. And last, we're delighted to welcome Rob Inaun, Executive Vice President, Global Head of Research and Development at Jazz Pharmaceuticals, nominated for Rylaze. So thank you to all of our panelists for being here. Really amazing work that went into each of those products, and that's what we want to hear about today. So let me start with a just a kind of an open question around how did you decide to start where you started? Why this disease? Why this indication? And in what way did your product, represent a breakthrough in this disease? And maybe I'll start with Gerald to kick us off.
Thank you very much, Michael. It's a privilege and great pleasure to be among such fabulous industry leaders today. So why did Daiichi Sankyo focus on a HER2 ADC? So many tumors are driven by particular mutations or overexpress certain proteins. In the case of the HER2 receptor, it's widely expressed in different tumor types, especially in breast cancer. So while we studied different tumor types in our initial study, we focused on breast cancer because so many women are affected by this disease, and about 20% of breast cancer patients have HER2 overexpressing tumors. And therapeutic options are limited, and frankly, the outcome is not so great. So the therapeutic breakthrough that Enhertu represents perhaps has two key aspects. One is the science behind the molecule, and the second is the extraordinary efficacy seen in the clinical trials. So regarding the science, ADCs, antibody-drug conjugates, bring a cytotoxic compound selectively to cells expressing the target, in our case, the HER2 protein. So Enhertu is the first ADC in Daiichi Sankyo's pipeline, and we have a very specific technology, I think, that represents a breakthrough. And it's characterized by a few things. We have a highly potent cytotoxic payload attached to the antibody, attached by a very stable linker that is selectively cleaved in the tumor cells. The cytotoxic drug has a very low concentration and short half-life in systemic circulation. And we have a desirable bystander effect. That means the cytotoxic component, after it's released from the antibody, can reach and kill neighboring tumor cells as well. And the other part of the breakthrough is just the unprecedented level of efficacy we have seen. So in our pivotal Destiny Breast 03 study in second-line breast cancer patients, we have seen that the median progression-free survival was tripled compared to T-DM1 or Kadcyla, which has been the standard of care for about a decade for these patients. This is pretty amazing. So yes, we addressed a clear unmet medical need, and we did it by a drug that is based on truly innovative science and has high efficacy. So that for us is a clear breakthrough, not just in breast cancer, but we have also seen that in other types of cancers.
Yeah, thank you, Gerald. I mean, just amazing technology. There's obviously lots of wonderful scientific detail you can get into in the choice of the antibody, choice of the payload, how you design the linker, all of which have tremendous degrees of freedom. But as you, I think, rightly pointed out, it really comes down to, and the impact that we've seen for patients is just startling. So maybe Lu Song, love to hear same question posed to you. How did you pick where you focused, and what kind of impact have we seen?
Yeah, so the time when we started our program, and that's at the very early stage of the program, we were fortunate enough to talk to some of the clinicians and found out that where the unmet medical needs are in targeting the heme malignancies, especially the B-cell malignancies. And that was the time actually ibrutinib started showing its promises in the clinic. And I mean, everyone knows that ibrutinib is a great drug, and the early sign it has in the clinic is fantastic. But then when we talk to the clinicians, and they noticed that even for a great drug as ibrutinib, and there were still some signals, and so at the time there were safety signals for ibrutinib. And especially the cardiovascular findings, such as atrial fibrillation. And in addition, there were hypertension and hemorrhage that were noticed. And so we had a hypothesis that this could come of the off-target effects. And so that got us to start and the journey to look for a better drug.
Kristen, would you mind going next?
Well, sure, thank you. So, it's a pleasure to be here. And I'd say BMS has always had a deep focus on doing research to identify innovative drugs for serious diseases such as cancer, and in particular, multiple myeloma, which is one of the more incurable blood cancers. And the story of the Abecma development, it starts in 2013 when BMS, formerly Celgene, established a partnership with Bluebird Bio. But really, that was not the beginning of the field in CAR T cells. It really dates back to the mid-90s, and I had the pleasure of being the first biotech company in this field back in the Bay Area in the 90s, doing the first clinical trials in HIV, actually, not cancer, and then solid tumors. And like many innovative cutting-edge fields, we learned a lot, but we weren't seeing the breakthrough results of today. And that company, went down during the recession of 2008. And followed the field until, around 2011, when you started to see these encouraging signs of activity coming with CD19 CAR T cells. And that's when I reconnected with one of my old colleagues from this little biotech company, and he was at Bluebird Bio, now 270 Bio, and they had a lentiviral vector gene therapy expertise, and at BMS and Celgene, we had a deep expertise in developing therapies for blood cancer. So we established this partnership in early 2013, really just around an idea that, we could work together to develop CAR T cells for cancers, and in particular, multiple myeloma, and see if we could extend the exciting early results with CD19 CAR T cells to a different target in a different disease. And, we can get into the sort of I call it the tsunami of drug development of that target from inception. But, I love the story because it was about an idea, jumping back and partnering on an idea, and then the opportunity to see that to fruition, to the breakthrough therapy that Abecma has been for patients.
Yeah, thank you. Another great example of modality innovation being a source of breakthrough, impact for patients. Not the only way to get that impact for sure, but obviously one of the key ways. Rob, would you take us home and talk about the story at Jazz and Rylaze?
Happy to. So as context, there was a big unmet need in ALL, acute lymphoblastic leukemia, for a novel asparaginase. Asparaginase is a key part of the multi-agent chemotherapy for ALL. It's known to contribute to the survival of patients who receive it, and it's one of the key reasons that survival in ALL is as high as it is today. The traditional way to give asparaginase is through derivation from E. coli, so asparaginase manufactured in E. coli. And when that's given, oftentimes there can be an allergic reaction. When an allergic reaction occurs, the E. coli-derived asparaginase is no longer effective. And so the solution for that for a long time was a drug called Erwinase, where the asparaginase is derived for Erwinia. And again, this is important for therapy because the malignant lymphocytes are exquisitely dependent on asparagine, so depleting asparagine is important. The challenge with Erwinase was chronic shortages. So oftentimes patients couldn't access the life-threatening drug that they needed. And those chronic shortages derived from the fact that it was very challenging to manufacture in a quality and reliable way. The analogy I use is it's like making penicillin in mold. That may be the way it was originally derived, but it's not the most modern way that would ensure quality and an abundant supply. And so Jazz undertook a process of evaluating alternative ways of manufacturing asparaginase that was derived from Erwinia, where there would not be a cross-reactivity for the allergy and could be given to patients who experienced an E. coli asparaginase allergy. And we settled on recombinant technology such that the Erwinia-derived asparaginase was actually made in Pseudomonas bacteria. And that turned out to be a very robust process that ensured the high quality and reliability of the product, and also a sufficient supply to ensure that all patients who needed it could get it.
Yeah, I love this example because it is proof of, there are multiple angles of innovation, all of which matter, right? Because what the end result is, are patients getting the care they need? And this is an example of a, kind of an orthogonal direction of innovation that people don't always think about, just how important it is to have a robust manufacturing approach. Maybe I'll reverse the order. So if you don't mind answering twice in a row, the second question is, okay, so we've had this impact. How have patients, seen it? What has their reaction been? How have physician groups reacted? And to the extent you feel comfortable, not everyone wants to answer or is positioned to answer these questions, but how have payers and access folks reacted? And I think we all saw this very provocative question at the start of this session, which I don't think any of us knew what the question was going to be, but it was around the Inflation Reduction Act and what that means for pricing and how we think about striking the balance there with payers around access. So maybe some of those subjects are a little far afield for folks in the R&D world, which is I think most of the panelists come from, but would love to hear your thoughts on reaction from these various constituencies.
Yeah, thank you. And so we've been supporting the leukemia community for many years at Jazz, both through commercialization of the prior product, Erwinaze, but also through other products we have. And so it was clear to us at the time that we made a decision to get into this scientific area that there were many patients and prescribers who were appropriately frustrated over the drug shortages. And what that often meant was that a patient who would have benefited simply could not access the drug because of the shortages. And so our development path was really closely aligned to input from patients themselves, but worked closely with the physicians who would ultimately be prescribers. And in this field, as you know, leukemia, it's a relatively small field internationally, especially on the pediatric side, where most patients receive their care through cooperative groups. So we were able to create a partnership early on that involved the Children's Oncology Group, which is the major pediatric cancer cooperative group in the US, as well as with the FDA, who was very interested in solving the problem of drug shortage and bringing a quality product to the market that could fulfill this unmet need. And so we worked with them from the very beginning around ways to develop the product in the way that would be most optimal for patients and to accelerate the timelines. And so there was a high level of interest from Children's Oncology Group, who were the participating centers in the clinical trial, to enable us to enroll the trial quickly, which in many cases was the only way for patients to access the drug during its development, and then to partner with FDA to ensure the review happened quickly. And the FDA actually used something called real-time oncology review to accelerate as much as possible the appropriate review of the safety and efficacy that ultimately led to its approval. So very high interest amongst all involved parties. And certainly since the approval of the drug now, really positive feedback from patients and prescribers, given the fact that it's broadly available when patients need it.
Kristen, how about you? Same question. Reaction from patient groups, from physician groups, from payers, market access folks.
Right. First, I think we're stepping back and saying, Abecma, which is a chimeric antigen receptor modified T cell, is a very unique and innovative therapy, right? It is made from the patient's own T cells that are genetically modified, retargeted to tackle and kill their cancer cells. And it's administered as a single one-time infusion. And it's a living drug. Those cells expand and can persist for months, even years. So if you take a patient population like multiple myeloma who have been receiving often continuous therapies for years, never with a break, the concept that you could get a single infusion of a therapy that could give deep, durable remissions, maybe not cures, that remains to be seen in multiple myeloma, but very long treatment-free intervals was really sort of groundbreaking and paradigm shifting. And as the early results for the Beckman came out, and I can still remember those early days in the phase 1 study when, patients who were, late line, had failed everything, came onto the study, their next step would have been hospice, they got the infusion of the CAR T cells, 2 weeks later, you do a bone marrow biopsy, the myeloma was just gone. You do a minimal residual disease assay, we couldn't detect the cells, less than 1 in 100,000 cells. I mean, it was really like nothing I'd ever seen before in my career. And, our investigators were passionate, our patients were passionate about it. We ran our pivotal trial in these very late line patients who had received many, many prior therapies. And, over 80% of them at the highest target dose in the study had a response, and almost 40% of those were complete responses. And if in the patients who achieved a complete response, their progression-free survival was over 20 months, almost 2 years. I mean, it was really unheard of. So, for us, the goal was how do we bring this innovative breakthrough therapy to patients as fast as possible? And then the next step is, how do we make sure, there's as much access as there can be to an innovative drug like this? And it's fair to say that to try to scale up manufacturing of autologous genetically engineered T cells is a challenge. It's a whole new area of medicine. We had to build new commercial manufacturing facilities. We had to recruit new cell therapy technicians, try to expand our capacity. And so, trying to match the incredible demand that there is now out there for CAR T cells like Abecma has been a major focus and a goal. And, we're still trying to increase our supply to meet that demand. And these are not inexpensive therapies, but, we think they're priced fairly based on the innovation and the value that they provide. And of course, at BMS, we have any number of patient access programs to help patients who are either underinsured or, challenged to access these medicines, gives them greater access. Across our myeloma portfolio, I think over 20,000 patients have benefited from these patient access programs. But you're right, I'm not a commercial patient access person, but it's been really gratifying for me from the research and development end to see, an idea be transformed into a product and now available to patients who, really desperately need therapies.
Yeah, thank you. I mean, there's parallels with Rob's story, right? Sometimes even the simplest things like logistics, right, end up being the thing that the treatment hangs on, and obviously true with CAR T, and also just great parallels to checkpoint inhibitors and the value of turning the body's own, cells against cancer and how durable the responses that you can get there. Great stories. Lusong, would you mind sharing with us, what you've seen in terms of reaction from patient groups and physicians?
Sure. Yeah, I think for patients and for clinicians, the most important thing is really the clinical data and the data for efficacy and safety. And so remember, we had the hypothesis that even as good as a drug as ibrutinib, and there is still room for improvement. And the hypothesis is to have a better, a more selective compound, and also just to have sustainable inhibition of the target in the disease tissue compartments. So those are the two aspects we try to improve. And it turned out that these efforts did translate into improved safety and efficacy in the clinic. And so in terms of the safety and tolerability, we did observe lower rates of cardiovascular events than ibrutinib, and that's about 3.5% lower. And that is based on the analysis about 780 patients who received zanubrutinib across 6 different clinical trials. And also there's a difference in the drug-drug interaction profile between Zanu and ibrutinib. And also Zanubrutinib can be taken without food, unlike ibrutinib, where a significant food effect was observed. And more importantly, on the efficacy side, and we did venture into running a couple of head-to-head phase 3 clinical trials against ibrutinib, and in Aspen and Alpine, the 2 trials, Zanubrutinib demonstrated superior efficacy and a more favorable safety profile than ibrutinib. And at the median follow-up of 19 months of the Aspen trial, both the clinical response and safety favored Zanubrutinib over ibrutinib. And in our recently announced final analysis result of the Alpine trial, Zanubrutinib achieved superior PFS versus ibrutinib in CLL. And so these actually provide potentially a practice-changing option for CLL. And so that's wonderful. So patients have more options. And in terms of access, and so we have always been committed to provide impactful and also affordable medicine around the world. So we're developing innovative and affordable medicines to improve the treatment outcomes. And so in the case of Brukinsa, Zanubrutinib, we already achieved approval in more than 50 markets, and that's in a very short time period.
Yeah, that's great. Obviously, making sure that patients all around the world get access is really critical. Gerald, I guess same question to you. You get to anchor us on this one. I mean, maybe you can tell us a little bit about the audience response at ASCO, which was extraordinary, but also just more broadly the physician, patient, and payer response.
Yeah, thank you, Michael. And I can only echo what you Song said, right? In different words, science is nice, but it doesn't matter if it's not bringing tangible, measurable benefit to patients. And in that respect, Daiichi Sankyo has been very fortunate within HER2. I mean, what speaks more to an amazing response from physician, and you alluded to that, than having thousands of oncologists get up at ASCO, the world's largest oncology meeting, and giving a standing ovation to Dr. Shanu Modi, who presented data, but also to the study, and this is the Destiny Breast 04 study, where we enrolled patients with very low levels of the HER2 receptor. So it was another scientific breakthrough, because we went there where all other targeted drugs have failed, and it took a lot of courage and daring spirit to go there. And the patients in that setting have only chemotherapy options with very limited outcomes. And what we have shown in that study that led to the standing ovation was a tripling of response rate, a meaningful improvement in the survival of patients. And it's not a small segment of patients. This is breast cancer, right? And what the outcome of that study is that approximately half of the patients with breast cancer now have potentially a better treatment option, and it's already approved in the US for that setting. And of course, another testimony to the importance of the data is that it was a simultaneous publication in the New England Journal of Medicine. And in fact, Enhertu now has 5 New England Journal of Medicine publications in less than 3 years in breast cancer, lung cancer, gastric cancer. I think that's a testimony to the enormous interest of the scientific community and the weight that the results have for patients, but also physicians. So the access question, pricing question is a very important one. And while everybody here on this panel is not an expert in market access and pricing, let me just say that I think for all of us as companies, this is a very critical question. For me, it is personally, as a medical oncologist, I've treated patients for many, many years. And we all know that governments face increasing budget pressures. So we have to come to reasonable agreements. I do believe that governments and health technology assessment bodies and insurers recognize the importance of innovative and potentially life-saving drugs like Enhertu, like others. And if they bring clear and meaningful benefits, and this is not just a better safety profile, this is really a tangible improvement in the survival of patients, then I think innovation will lead to an adequate, I think, pricing of the drugs.
Yeah, well said. I mean, look, all of your companies spend billions of dollars every year in R&D, and that is that kind of investment. It only happens if there is at least some financial return. Obviously, there's also a return to patients that companies focus on, but it's hard to justify if there's not adequate financial return. And so, ensuring that happens when for every drug that makes it to market, there are hundreds that failed somewhere along the way, is obviously part of the equation here. And maybe that's the next question I'll turn to. And I won't ask everyone to answer it. And well, you all can if you want to, but I'll open it up, which is, let's talk a little bit about that difficulty of getting from an idea to actual impact. We use the word translation, in the industry, meaning how do we go from early idea, something maybe we've seen in some animal models, maybe even some initial human data, but not yet proof of concept. How do you get from that to, yes, I know I have a therapy that I know is going to work. I've got sufficient data to show that, and now it's on the market. And talk about bridging that translational gap. I wonder if any of you would like to kind of talk through your own story in that. It doesn't have to be just with this medicine, obviously, because there are many other assets that I'm sure you've been affiliated with that maybe haven't made the leap, and you can talk through some of the challenges there as well.
So Michael, if I could just add, to the previous comment also you made, right? And it ties also to the pricing and market access question, but of course to the challenge to bring something from, the bench to patients. In the case of Enhertu as our Daiichi Sankyo's drug, we had roughly 4 years from the first patient receiving the drug to the first approval. So we had to make a very bold and courageous decision to invest heavily in the production of the molecule. And that means setting up facilities, and antibody-drug conjugates are very expensive. So the company had to invest a couple $100 million at risk, not knowing if this would ever lead to a drug that can be approved. And it's not the cost for the studies, it's just for manufacturing of the drug. So that is clearly one of the challenges that we have if we bring things from a bedside to the patients. And another comment that maybe applies also to the other panelists here. If you have early studies that show very strong efficacy, then you have to do confirmatory studies or larger studies where you randomize against a control arm. And patients today read about everything they can find, and they know about early efficacy of drugs. So to keep patients that are randomized into a control arm of a study in that control arm has turned out to be a big challenge for us, and I think for many of us. So a lot of challenges, and just to highlight two of them.
Yeah, it's Chris, and I'd love to build on that. Focusing just on the manufacturing. You have traditional off-the-shelf pharmaceutical products that you manufacture at scale at store, and then you can just deliver to patients whenever they need them. And then you fast forward to the bleeding edge of scientific innovation, where you're collecting fresh T cells, from patients around the world, bringing to a facility, infecting them with a virus, growing them in a culture dish, freezing them, testing them, releasing them, shipping them back to the individual patient. And you can imagine, that was an early field, right? The CD19 field was just kicking off as we were developing BCMA CAR T cells, and as soon as those first patients started to respond, you realized we need to take that little manufacturing facility that was designed for a phase 1 study, and now we need to build a commercial CAR T cell manufacturing plant. We need to figure out how to collect and ship these cells from all over the world, manufacture and get them back. One of my favorite stories was in the phase 1 study when there was a big snowstorm in New Jersey. And, there were CAR T manufacturing processes ongoing, and our technicians put on their skis and they skied to the manufacturing facility, and they called their friends, the police officers, to make sure they didn't tow their cars off the road because snowplows were coming through. But, they knew they were making a product for an individual patient. And so, everyone was going the extra mile. And when we were building that first commercial facility in the middle of our phase 1 study, we told all the contractors what they were building. And Tom Whitehead, the father of Emily Whitehead, came and gave them a sort of a talk. Not a single person took a day off. And we built that facility 6 months ahead of schedule because everybody knew they were building, treatments for cancer patients, for individual cancer patients. So, there's full, all of this is full of setbacks, and you're, as they always say, you're building the runway while you're flying the plane, and you pivot and you have surprises and you tackle them, pivot again, and maybe that's why I like early drug development. But you amplify that with the race to get a product approved and go from that first evidence of efficacy in a phase 1 study to an approval of a complex autologous therapy that can be delivered on a global scale. And you just can't just underappreciate what a massive effort that is. You don't want to draw parallels to COVID vaccines, but when you see what's possible driven by the promise, it is amazing how fast and efficient our field of biopharma agency can be in this, line of sight to get something done. And sometimes when you're frustrated that things aren't as fast as you hope they could be, you can look at these examples and see what's possible. So for me, that's what's been very exciting. And I hope people out there understand, the incredible complexity involved in bringing these kind of therapies forward to patients, but also the incredible commitment and passion on part of the people that are doing this.
It's a fantastic story and an amazing image that will stick with me of, skiing down Route 1, to get to work, literally going the extra mile, as you said. I think you raised a very provocative question, which is a COVID devastating disease, and we had to be all in to answer it quickly. But the reality is there are as many other diseases that, kill hundreds of thousands a year. Those patients are very much in need as well, right? And can we bring some of the urgency that we all felt around COVID to all these diseases across the industry, regulators, public at large around this? So I thank you for raising that. Lusong, Rob, do you have thoughts on this, getting across the translational gap, Valley of Death?
Sure, I can start. And so we were a little bit different in terms that when we started our clinical trial for the zanubrutinib, we were a very young company, 3 years old. And so you can imagine the challenges we're facing at that time. So everything is difficult for us. We don't have the resource. We don't have billions of dollars to spend in R&D every year. And so we were learning along the course. And especially if you think about bringing in a head-to-head trial against ibrutinib, and so how daunting that task is, and even just to secure the competitor drug, that's a huge challenge. And so, what we learned along this process is we have to believe in the science, and we have to believe in the people, and also when the time is tough, and we have to really keep in mind why we are doing this. And so it's really for the patients and really to address the unmet medical needs. I think this probably is true for any of the efforts we are making in this industry.
So Mike, the challenge for us was a little different in that, as I described earlier, we set out to kind of solve a technological problem, to make an asparaginase that was essentially, from a biochemistry perspective, identical to what was already on the market, but to improve the process around quality and reliability and scale up. And we knew from the preclinical experiments that the activity level was right on target to the reference standard. And even from the initial healthy volunteer study that it was as well. As we then took the next steps to evaluating patients, we began to understand better that the field had moved on a bit. And in our collaboration with Children's Oncology Group and with the FDA, there was a clear desire to improve on the outcomes from asparaginase therapy over what had been the standard with Erwinase. And in particular, to ensure that more patients had more optimal reduction in their asparagine levels through the dosing interval. And so essentially, because the literature was showing the importance of essentially minimizing asparagine to malignant T cells over that entire dosing period, we then had the challenge of not only making the drug that was higher quality and more easily produced, but to then explore different doses that could optimize it for the patient as well. Fortunately, the biomarkers in this space are really very good and predictive. And so we could do that dose ranging by simply measuring things like asparaginase activity in the blood or the extent of depletion of asparagine in the blood. And so we leveraged that ultimately to not only demonstrate that the new drug had the same activity as the reference standard, but to provide data on a better dosing paradigm that would ultimately give better results for patients. And so that was the translational challenge we had. Ultimately, we were able to solve that through iterating through a number of different doses and schedules and doing the population PK modeling that would allow us to minimize the number of patients to get to the most accurate predictions.
Yeah, it's a great story, and I'm sure, obviously, in your role, leading R&D, right? There's lots of examples of other situations where you've also, different parameters of getting across that translational gap come to bear. Let me ask kind of another open question, and actually very prophetic version, which is put on your prognosticators hats. You're all experts in this area. What are the new innovations that we can look forward to over the next decade, let's say, that are going to shift the way we think about care? And we'd love to hear your what's in your crystal ball. And again, anyone and everyone can answer this.
Well, I maybe the first part of my answer would just be, I've really enjoyed this session because, of course, I already knew quite a bit about the products being discussed today, but to hear some of the details firsthand from the innovators just makes you really optimistic about the future. I think that's probably, the genesis of your question. And so as I think about, what's resulted in, or at least what the, let's say, the enablers of the innovation you've heard about, it really has to do with a deeper and deeper understanding of the diseases that we're all studying. What drives cancer, what prevents the immune system from managing cancer, what the targets would be for other diseases. And given the foundation of our science has evolved so rapidly in the last few years, there's been the opportunity to develop the drugs that you've heard about today. And I'm just very optimistic that that will continue. And in 5 years, where we have the same panel, you'd be hearing about a whole new slate of additional innovations.
Daryl?
Yeah, I could only agree to Rob. I think we had in the past maybe waves or decades, which certainly overlapped, after the era of chemotherapy, there came the kinase inhibitors, there came anti-angiogenic drugs, then immuno-oncology and cell therapy. Now we may have a decade of ADCs, but I think it will all converge. And as oncologists, we always need to use an armamentarium of different therapeutic options. So I think it may not be one specific breakthrough. It may be a continuation of finding additional tumor-specific targets. maybe specifically and more intensely targeting the tumor microenvironment, which I think does not yet play as important a role as it should be. I think earlier detection of resistance and targeting resistance mechanisms is important, and ultimately sticking to what I'm doing in Daiichi Sankyo is improving antibody drug conjugates, because if we have better technology to bring drugs specifically to the cancer cells, we can potentially improve the outcome of our patients. So I can only echo, I would be very exciting to dial in 5 or 10 years from now and see what additional innovations and scientific advances our patients will be able to receive.
Yeah, I guess I would say, similar to everybody else, just a general level of excitement about what's to come. And part of that will be informed by the translational learnings of today. So, for example, at BMS, we're the only company that has 2 approved CAR T-cell products against 2 different targets and 2 different diseases. We've treated thousands of patients now. And all of that translational data is available to interrogate and inform the next generation of cell and gene therapies, and hopefully allow that extension from what have been breakthrough therapies in blood cancers to the more common solid tumors. And we have lots of investments with small biotech companies and internal discovery efforts to look at different effector cells, NK cells, gamma delta T cells, different sources of those cells, induced pluripotent stem cells, different engineering tricks to increase the fitness or the function or the persistence of these cells in patients. And just the amount of research happening in the cell and gene therapy space today compared to when I was at the one company in this field, right? There are hundreds of companies in this field now. You can only imagine the innovations that are going to come. And it's well beyond cell and gene therapy. You've heard about next generation antibody drug conjugates. You can imagine that field could blow wide open with novel payloads, for example. And then you've got, the complex biologics like the bispecific immune cell engagers, very exciting data with these T-cell engagers. We're now exploring NK cell engagers. And then the many, many ways that you can modulate the immune system to help fight cancer, not just the effector side, the T-activated T cells, but the suppressor cells like the Tregs or the myeloid suppressor cells. So, that's an exciting field. And then beyond that, if you step aside from cancer immunotherapy, there's a lot of excitement these days around targeted protein degradation. We have a big discovery effort there as well, building off our legacy of IMiD drugs and cereblon modulating drugs. And rather than inhibit a kinase, if you can target and degrade a critical protein that's required to maintain the cancer state, that's a whole new angle on treating cancer. So, the next decade is going to be incredibly exciting. The tools and the technology and the translational learnings from today are really hopefully going to inform a much faster innovation to come.
Yeah, thank you. And yeah, the proteasome ubiquitin degradation is super interesting. We'll see where it goes. Lusong, would love to have you kind of close us out with your crystal ball, and then we'll open it up to the audience for questions. And I'll just ask the Galien folks to make sure we can see those questions, because so far I don't see anything on my screen yet, but hopefully they'll start popping up. So if you have questions, please start inputting them so we can get them loaded. But Lusong, please.
Sure. So I think there are many different layers of innovation we're looking at. So one observation I had today is, so for all 4 drugs we're talking here today, and none of them are the first approved for that target. But however, I think there is a lot of innovation here and a lot of improvement. And I would say sometimes tremendous improvement, and offer patients new options and a lot very meaningful options. And I think that could, through the new modality in terms of CAR T versus the other modalities, or better compounds and a better technology with the improved ADC and all that. So I think that is probably traditionally what people would say that's best in class opportunity. And that's very meaningful innovations. And then there's a next layer, I would say, would be first in class, and there are a lot of undruggable targets out there, and there are just not options. And so if you go to do your mutation sequencing, and then you find P10 mutation or some of the other mutation, APC, beta-catenin, and there are just not many options out there. But I think with a lot of the breakthrough in the technology, with the PROTAC, with our mRNA therapeutics, with the novel antibody, small molecule, or other conjugates, I think we're going to see more and more options out there for the patients, and we are going to see more of the promises coming out of the clinic and address those on medical needs or undruggable targets before. And we've seen that through the years, especially in the past decade. And I would also say that there's another layer, and I think that's probably at the end of the future, just as in the infectious disease in oncology. I think combination is going to be more and more important. And so we are start seeing the combination of the IO, among IO, so IO target therapy or IO and chemotherapy. And I think combination is probably going to really, really give us meaningful survival benefit at the end. And I think that really demand or call for a very safe drug and a very deliverable drug that can really treat cancer as a chronic disease.
Yeah, it's a great point, and Gerald made the same one. And look, cancer cells evolve, and if you make the analogy to infectious disease, which is the pathogens evolve, and see what we've done with heart and HIV therapy, right? There clearly is a logic around hitting something multiple ways simultaneously to really try and suppress it. So I'm looking for audience questions. It may be that I just have an issue logistically with not seeing them pop up, but so if the Galien folks, if you can make sure that the questions are coming through, if they are being put forward. And audience, if you have questions, please, no, there's no such thing as a bad question. So please, share your thoughts, because that's always, it's always fun to get something that we maybe haven't thought about on our own yet. So please put your questions in. And while we're waiting, just to see if those two come through, maybe one more question for me, which is, so as you kind of think about the advances that have been made in cancer, I sort of alluded to it, right? That there's a little bit of the way ideas can move between disease areas or therapeutic areas. Are there lessons that you would take from oncology? I know I'm asking you to get out of your lane here, but are there lessons you would take from oncology that you think are important insights for other therapeutic areas? So for instance, a big advance in cancer was when we started to be able to genotype tumors and understand really the mechanisms that were driving various transformations, whether it was angiogenesis or turning growth on through growth factor receptor overexpression, et cetera. There are other areas where maybe we haven't got as deep of an understanding of the biological basis yet, in neuroscience, and you can pick a few others, maybe even in immunology, it's starting to emerge. Are there lessons you would share that you think might be important in other therapeutic areas?
Yeah, I would take that. If you just think of the flip of the coin between cancer immunotherapy and autoimmune disease, where, for cancer immunotherapy, trying to turn on the immune system to target and kill cancer, but for autoimmune disease, you're trying to shut it down. And I'd say one of the lessons from oncology that we're trying to apply to autoimmune disease is that more in-depth mechanistic understanding of what causes cancer. And within any given cancer, lung cancer, right, there may be 20 different drivers, different subsets, and take that kind of mechanistic insight, targeted therapies for the right subset of cancer to target the driver mutations, the driver inhibitory aspects of the tumor microenvironment, and translate that to autoimmune disease, where it's not just lupus, where it's not just MS or rheumatoid arthritis. There are likely different subsets with different mechanistic drivers that would be most amenable to different therapeutic approaches. So I think that kind of mechanistic disease model-based understanding, where I think cancer is ahead of the curve, and to take that mindset, a lot of tumor biopsies, a lot of tissue biopsies, to try to inform and select the right patient for the right treatment, right, the right disease.
Like, maybe the other thing I would highlight is that, and I know this is true for many other areas, but certainly in my experience in oncology, there's been a really close partnership between patients, patient advocacy groups, regulators, industry scientists, clinicians, and academic scientists around really accelerating development where it's appropriate to do so. And I do think we are seeing that in other fields as well, but maybe we could even see more of that. You certainly saw it with COVID vaccines, where it was clear that you needed to accelerate the approval to derive the maximum benefit, and the benefit risk was there pretty early on. And I think some of that collaboration could be further leveraged around, say, accelerated approvals, et cetera, in disease areas where there's a high unmet need and a strong rationale to do so.
Yeah, I would like to agree with Christian, and I think the analogy between oncology and I&I, and also the biomarker approach is really important, and I think it's probably going beyond just the inflammatory disease, and I mean, even in metabolic disease and other diseases, and you just don't see many practices just like oncology we're doing today, and where it's pretty mature and common now to go with a biomarker driven approach. And so I think it'll be a very, very good practice to extend beyond oncology.
Yeah, that's a great point. So we're almost at the end of our session here. So I'll ask the Galien folks to put up the results of the survey. I think that's what you do now, but correct me if I'm wrong on that. But I'd be curious to see what the poll results were. And then while we're waiting for that, I just wanted to share a couple of logistics for folks. First of all, I want to thank our panel. I tremendously enjoyed that discussion. I learned a lot. I think Rob, you probably captured it very well, or just like listening to each person kind of walk through the story was incredibly eye-opening. So thank all of you. There will be a webcast of this available if you just go to the Galien Foundation homepage. The Thursday meeting during the Jubilee Forum, there's a group CEO keynote followed by a presentation of the 2022 Roy Vagelos Pro Bono Humanum Award. That will be at 6:30. And then there'll be the Gala Awards dinner, both of which are held at the American Museum of Natural History. We can react to it. Otherwise, I'd like to just thank everyone for joining.